Joe Lewis, European Molecular Biology Laboratory. Small molecules with anti-cancer action.
Okay, good afternoon. Christian, I don't like Lebkuchen, but I do like whiskey. Okay, fantastic. So what I want to do today is tell you about some of the work we're doing up at EMBL on RNA binding proteins as novel targets in oncology. And as many of you know, misexpression of RNA binding proteins has very profound effects on cellular metabolism and cancer pathology in particular. The protein we are particularly interested in is called FXR1, and it's found amplified in many different cancers and is associated with 3q26 Amplicon. And it's known that knocking down of FXR1 can stop tumor growth in vivo and also stop cell proliferation and invasion. The protein itself is involved in translational regulation of a number of different genes, including tumor suppressors, and this is how its mechanism of action works. The 3q26 Amplicon is great because it actually gives us a biomarker to identify patients that could potentially respond to our small molecules. What you see here is in squamous cell carcinoma of the lung, over 50% of the patients have this Amplicon, so FXR is amplified, whereas in contrast in adenocarcinoma, it's less than 5%. And in the Kaplan-Meier curve, what you can see is overexpression of FXR1 actually correlates with a poor clinical outcome. So being able to reduce the activity of FXR1 should have a positive clinical benefit. But in order to identify small molecules that can actually modify FXR1, you need an assay system, and this is shown in overview here. Basically, we have a messenger RNA where the translation is inhibited in the presence of FXR1. When we have a small molecule that inhibits its activity, we get translation, we get signal, we can use that to measure. Using this, we've identified small molecules and developed them to mid-lead optimization. And the most advanced of these we've evaluated in lung tumor xenografts that overexpress FXR1, and this is what you see here. If you look at the bottom curves, you can see that 625 is just as active in this in vivo model as a standard of care carboplatin, which is a very good place to be. So just to summarize, we've got 2 independent chemotypes at the moment that we're actively working on. We're validating this target chemically at the moment. And I think what you saw is very good in vivo activity in mouse models. And I think you'll agree with me, it's a very good starting point for clinical development for a novel target class. Thank you.